Welcome to G Fun Facts Online. Today, we're embarking on an odyssey into the eating habits of our ancestors. When you picture a prehistoric meal, you probably imagine club-wielding cavemen gnawing on giant mammoth steaks. But the real story, pieced together by the science of paleodietetics, is far more complex, varied, and fascinating. This isn't just about curiosity. It's about understanding how diet shaped our very biology, our societies, and our planet. Scientists are rewriting the story of what it meant to eat and survive hundreds of thousands of years ago by analyzing clues from fossilized bones, teeth, and even ancient feces. So let's explore the fossilized cookbook and see what our ancestors were really eating. The most direct evidence for meat consumption comes from the bones of prehistoric animals. Fossilized bones with distinct cut marks, left by sharp-edged stone tools, provide a visceral connection to our ancestors' carnivorous habits. These marks, often found near joints or areas with lots of muscle, show that hominins were systematically processing animal carcasses for meat and marrow. But how can we be sure these marks are from tools and not, say, a lion's tooth? Researchers use powerful microscopes and 3D imaging to analyze the shape of the incisions. Stone tools typically leave a V-shaped groove, while a carnivore's tooth leaves a U-shaped one. Marks from being trampled underfoot are usually shallow and curvy. Some of the earliest, though debated, evidence of butchery dates back an astonishing 3.4 million years, with cut-marked bones found in Dikika, Ethiopia. This predates the genus Homo, suggesting our earlier Australopithecine ancestors might have been using tools to get at meat. By about 2.6 million years ago, with the first definitive stone tools, the evidence for butchery becomes widespread, showing that meat was a regular part of the hominin diet. While bones tell us about the meat, teeth tell us about everything else. Teeth are the hardest part of the skeleton and are often exceptionally well preserved, making them a treasure trove of dietary information. The hardened plaque, or dental calculus, that clings to ancient teeth is like a microscopic time capsule. As this plaque mineralizes over a lifetime, it traps an amazing array of microscopic food particles. Scientists can extract and analyze this calculus to identify tiny starch grains and phytoliths, which are silica structures from plant cells. This can reveal the specific types of plants consumed. For example, analysis of Neanderthal dental calculus has found starch grains from grasses, legumes, and even date palms. Crucially, some of these grains show damage consistent with cooking, which directly challenges the old idea that Neanderthals were exclusively meat eaters. The emerging field of paleoproteomics even allows scientists to identify specific proteins preserved in calculus. This has provided direct evidence for the consumption of milk products and cereals, foods that don't leave big fossils behind. And sometimes, even ancient DNA from plants and animals can be recovered from this calculus, allowing for incredibly specific identification of food sources. Beyond what's trapped in the plaque, the tooth surface itself tells a story. The act of chewing leaves a microscopic landscape of scratches and pits on the enamel, a pattern called dental microwear. Harder foods, like nuts and seeds, tend to leave more pits. Softer foods, like leaves and fruits, create fine scratches. By comparing the microwear on fossil teeth to modern animals with known diets, scientists can infer what our ancestors were eating. For instance, the hominin Paranthropus boisei had a massive jaw and huge teeth, which led scientists to believe it ate very hard foods. But microwear analysis showed mostly light scratches, suggesting a diet of softer foods, with tough items possibly being fallback options during times of scarcity. Now for the most direct and perhaps least glamorous evidence of all: fossilized feces, or coprolites. Coprolites offer an unparalleled window into the actual foods an individual consumed and digested. Inside them, we can find undigested food remains like bone fragments, hair, fish scales, seeds, pollen, and plant fibers, giving us a clear picture of last meals. For example, coprolites from Hines Cave in Texas, dating back over 6,000 years, showed that early inhabitants ate a huge range of plants and animals, including cactus, walnuts, agave, and even small rodents. Coprolite studies have also been key in demonstrating the high-fiber, plant-based nature of many ancient diets, with significantly higher fiber intake than most modern diets. They can even preserve intestinal parasites, giving us insight into ancient health, and a DNA analysis of coprolites is beginning to reveal the composition of the ancient human gut microbiome. Beyond what we can see, a suite of powerful chemical techniques allows scientists to delve even deeper. The most important of these is stable isotope analysis. The principle is simple: you are what you eat. The isotopic signatures of the foods we consume are incorporated into our body tissues, like bones and teeth. By measuring the ratios of different stable isotopes of elements like carbon and nitrogen, scientists can reconstruct an individual's long-term diet. The ratio of carbon-13 to carbon-12 reveals the types of plants at the base of the food web. Plants fall into two main groups based on how they do photosynthesis. C, 3 plants like trees and shrubs, and C, 4 plants like tropical grasses. By analyzing carbon isotopes in a fossil, scientists can tell if their diet was based on C3 or C4 plants, or the animals that ate them. Nitrogen isotopes, on the other hand, indicate an organism's position in the food chain. Nitrogen-15 accumulates as you go up the food chain, so herbivores have lower levels than carnivores. High nitrogen-15 values in Neanderthal bones, for instance, are similar to modern carnivores, pointing to a diet rich in meat. Stable isotope analysis has been revolutionary, revealing a major dietary shift in our ancestors after 4 million years ago towards eating more C4 plants or the animals that ate them. It also shows that many hominin species were incredibly flexible, with different individuals eating different proportions of these foods. So, with this impressive scientific toolkit, what was actually on the prehistoric menu? Let's start with our closest extinct relatives, the Neanderthals. For a long time, they were seen as top-level carnivores. And it's true, isotope analysis shows high nitrogen levels consistent with a meat-heavy diet. Archaeological sites are full of the remains of large game like mammoths and reindeer, with clear butchery marks. In coastal regions, they also ate mollusks, seals, and fish. But a growing body of evidence shows Neanderthals were not just meat eaters. That dental calculus we talked about has been a game changer. It has revealed microscopic remains of a variety of plant foods, including grasses, legumes, and tubers, from Neanderthals in both warm and cold climates. Many of these plant remains show damage from cooking, suggesting Neanderthals had a sophisticated understanding of food processing. In one incredible case, DNA from poplar bark, which contains the active ingredient in aspirin, and traces of the antibiotic-producing Penicillium mold were found in Neanderthal dental calculus, suggesting they may have even used plants for medicine. Coprolites from a Neanderthal site in Spain also confirm a diet that included nuts, berries, and root vegetables. The picture is clear: Neanderthals were adaptable omnivores. What about our own species, Homo sapiens? We appear to have been characterized by even greater dietary flexibility, adapting our menus to whatever environment we inhabited. At a 100,000-year-old cave in Mozambique, there's evidence for an extensive reliance on wild cereals like sorghum, as well as fruits, nuts, and tubers. This is the earliest direct evidence of humans using pre-domesticated cereals. At a 780,000-year-old site in Israel, starch grains from acorns, cereals, and aquatic plants were found on stone tools, again challenging the narrative of a purely meat-based diet for early hominins. More recent studies from the Andes, on remains from 9,000 to 6,500 years ago, suggest that plants, particularly wild tubers, made up as much as 80% of the diet for some early hunter-gatherer groups there. And cooking these starchy foods seems to be a very ancient practice. Charred pieces of tubers from the Klassies River site in South Africa date back 120,000 years, making them the earliest known evidence of our species cooking carbohydrates. This ability to unlock the nutritional potential of starchy foods would have been a massive advantage, providing the energy necessary to fuel our large brains and support our migrations across the globe. Of course, our ancestors also hunted giants. The Pleistocene epoch, or the Ice Age, was a world of megafauna: mammoths, mastodons, giant sloths, and woolly rhinos. There's clear archaeological evidence that early humans hunted these massive animals. Paleo-Indian sites in North America frequently contain mammoth remains alongside distinctive Clovis spear points. Hunting such large animals would have provided a huge caloric payoff, but it was also very risky. There's a long-running debate about what caused the mass extinction of these megafauna at the end of the Pleistocene. The overkill hypothesis suggests that the arrival of skilled human hunters was the primary driver. However, rapid climate change was also a significant factor. Today, most scientists agree it was likely a combination of human pressure and a warming climate that led to the demise of these magnificent creatures. So what is the enduring legacy of these prehistoric meals? The science of paleodietetics shows us that there was no single paleo diet. Instead, our ancestors were remarkably flexible and opportunistic eaters, thriving on a wide variety of plant and animal foods depending on where and when they lived. This dietary adaptability was the key to our evolutionary success. The ability to exploit a diverse range of food sources allowed our species to survive and flourish in nearly every environment on Earth. This research also challenges many modern fad diets. The popular notion of a meat-heavy caveman diet is simply not supported by the diverse archaeological evidence, which increasingly points to the critical role of plant foods, especially cooked starches, in human evolution. Our evolutionary history is not one of dietary specialization, but of incredible culinary flexibility. As we continue to refine the tools for peering into the deep past, the story of what made us human will undoubtedly become even more detailed. The fossil record is a cookbook millions of years in the making, and we are only just beginning to decipher its recipes. Thanks for listening to G Fun Facts Online.